Hi all, welcome to our second bioinformatics online seminar this semester. So I'm glad to take the privilege to introduce our great speaker today. So Dr. Geyu Zhou is an assistant professor of biology and statistics at Purdue. So he received his PhD degree in computational biology and bioinformatics, followed by a postdoc training in biostatistics at Yale. So he currently works on genetic prediction, admission mapping, and gene by environmental analysis in mixed populations. His work is supported by the Analytics in Fellowship. So for our assignment today, we will take questions at the end, but feel free to post your question in the chat during the talk, and please remain muted throughout the talk. So now the floor is yours, Dr. Zhou.
Okay, thank you. Very nice introduction, Lauren. So today I'm going to mainly talk about my research on the genetic risk predictions, and my title is the Bayesian transfer learning with 2D derivative process random effects model. So before talking to the statistics, let's talk about something about the biology. As we may know, for the human genome, we have 22 pairs of autochromosomes and a pair of the sex chromosomes. And on chromosomes, there's DNA with the genetic information coded in four base A, T, G, and C. So the human genome has about 3.2 billion base pair, which codes about 20 to 30,000 proteins. And we are mostly interested in studying the genetic variations. We know that 99% of the human genomes, they are same across individuals, but for 1% of the human genomes, there contains genetic variations that really determines why I am different from you. And generally speaking, there are two kinds of genetic variations, the single nucleotide polymorphism, the SNPs, or the more complicated structural variations. So for the single nucleotide polymorphism, it is just a change of the base pair at a single nucleotide level from the reference. For example, I may carry the allele A here, and other people may have the allele like C or G. Structural variations are more complicated. It typically involves more than change of the more than one base pairs. For example, we have deletions, insertions, or duplications, inversion or translocations. In today's talk, I'm going to mainly focus on the study of the single nucleotide polymorphisms. So one thing, when we have understand of the genetic variations, one thing we can do is to really dissect how those genetic variations contribute to the human traits. So human traits can be generally divided into two categories. The Mendelian traits, which typically follows the Mendelian inheritance, such as autosomal dominance or recessive inheritance patterns, and they are typically controlled by mutations in a single gene with rare, but often rare, but with large effects. Some examples of the Mendelian traits are cystic fibrosis, which is caused by CFTR gene, and Huntington's disease, which is caused by HTT And more traits, they are complex, which means they do not have a very simple Mendelian inheritance patterns. This means these traits, they are often influenced by many genes with also the interactions and effects from the environment. And for these complex traits, typically each genetic variant only has a tiny effect size compared with the Mendelian traits. Some examples of the complex traits are high blood pressure, lipid level, type 2 diabetes, and schizophrenia. And to study how genetic variation is linked to the complex traits, people often carry out so-called genome-wide association study. So genome-wide association study, or GWAS, works by first recruit the cases and the controls to form a cohort. And then it will genotypes a bunch of the SNPs across the genomes, and then going to study what variants or SNPs they are associated with diseases or complex traits. And this is what a GWAS data looks like. Suppose we have three people and we have the genotypes at three SNPs. And for the person one, for the SNP, there's AA, and for other people and other SNPs, we can similarly read the genotypes. And suppose if for the SNP1, we treat A as the effect allele, then person 1 carries two copies of the A, so we can code the number as 2, and person 2 carries one copy of A, so we can code as 1, and person 3 carries 0 copy of A, so we code as 0. And we can perform a regression of the disease status of each person on the number of the alleles carried by person at each SNPs to obtain the effect size and the p-value from the regressions. So the effect size and the p-value will measure the strength of the associations between the SNPs counts, the risk allele counts, and the disease. And typically, we have millions of SNPs being genotyped and analyzed in this way. And after we obtain the results, we can plot each SNPs on the chromosomes ordered by their positions. And on the x-axis and on the y-axis, we can plot the minus log 10 p-value. And typically, we will see if there's genetics factors associated with the trait, we will see there's lots of SNPs are reaching the genome-wide significance level. And here we see a little bit peaks on the plot. This is because SNPs nearby each other on the chromosomes, they are typically in linkage disequilibrium, which means their SNPs, they are highly correlated. So if we do a test on two SNPs that are correlated, their testing statistics will also be highly correlated. So if one of them is significant, the other is also highly likely to be highly significant. So that's why we will see there's some peaks on the plot. And this is also why this kind of plot are often called as a Manhattan plot. So now I'm going to talk about my research, which typically deal with the genetic risk predictions. So in 2012, people came up with exciting ideas to see whether we can construct a prediction model based on the genetic variants and the informations from the GWAS study. The idea is promising because most of the genetic variants, they are germline, which means they do not change across a lifetime. So if we are able to construct a risk prediction model, it can be applied earlier in the life to inform a prevention strategy. And indeed, there are companies like 23andMe and Ancestry. They are very excited about this kind of predictions and actually offer commercial service to do this. And today, I'm going to mainly talk about how we are able to construct such prediction model. So one simple approach is we start with results from the GWAS data and use it as a training data set to construct a model. The way it does is we simply take out the SNPs, the effect size, and the effect allele from a GWAS results. Then for a new individuals, we measure the counts of the effect allele at these three SNPs. For example, at SNP1, the read is AG, so it carries one copy of the effect allele. And SNP2 is AA, so 0 copy of the effect allele C. And at SNP3, GT, so one copy of the effect allele G. And then we multiply the number of the effect allele by the effect size and take the sum to get the final score. And this is so-called polygenic score. So why is it called as polygenic? It basically means the effect size of each variance is typically small and the number of SNPs, they are large. So we need to aggregates thousands of millions SNPs across the human genomes to formulate our prediction model. And that's why it's called polygenic. A central question to ask is how accurate are those prediction models? So the predictions highly depends on the accuracy of the estimation of the effect size as the genotype read is fairly accurate from our current technology. And in 2018, there's a paper published by studying the genetic risk scores calculating of populations in UK, and they look at the people with coronary artery disease and without coronary arteries, and they compare the polygenic scores in case and versus controls, and they are able to show that in the cases, the polygenetic score percentile is significantly higher than the controls. So this offers a potential value to identify those cases based on the score. And more recently, earlier this year, there's like among one of the first clinical trials being conducted to evaluate the efficacy of the polygenetic score in screening for prostate cancer. And they found that for prostate cancer, the polygenetic score offers additional clinical significant values compared with the traditional screening approach, such as a PSA or MRI. So this basically means it has some potentials or promise to translate a polygenic risk prediction model into a clinical practice. And now I'm going to talk about the statistics behind how to construct such a prediction model. So all the GWAS models, they are based on the linear regressions y equals x beta plus epsilon. And y is a vector of phenotypes with n individuals. And the genotype is typically a n by p matrix with n as the sample size and p as the number of SNPs or markers, and beta is a vector of the genetic effect size to be estimated, and epsilon is a random effects. And once we got an estimate of the effect size beta, and we obtain an estimator beta hat, we can construct the prediction models in new samples by simple linear sum. There's one challenge of fitting this kind of linear model because the number of the SNPs or genetic variants is typically much larger than the number of individuals and the sample size. So there is a high dimensionality problems, which means we cannot use the ordinary least square to fit the model due to the rank insufficiency of the X. And typically, people prefer to perform some regularization and impose the shrinkage to avoid the overfitting. In the traditional statistics or machine learning literatures, there are frequencies approach such as lasso, ridge regression, or elastics that can be helpful to build such a machine learning prediction model. But in this field, people now heavily favor for the Bayesian approach, which means we put some prior assumptions on the effect size of the beta and do the Markov chain Monte Carlo to get the posterior estimate of the effect size. And perhaps one good thing about the Bayesian approach or the advantage of Bayesian approach is it really allows for a more flexible assumptions compared with the traditional approach, as I will show later. So for the Bayesian prior, you can really see the different methods makes a different assumptions about the Bayesian prior. For example, the simple way is to just assume the beta follows a single normal distributions. So this is also called blob or the linear mix model. And lasso or lasso sum, they will put a L1 penalty on the effects. LD2 assume that the beta follows a mixture of the normal distribution with probability pi being non-zero and probability 1 minus pi being zero. So this is also often called as a spike and splat prior in statistics, which means only a part of SNPs will have effects as non-zero. And the BSLM or DBL is the same. They all assume that the beta follows a mixture of two normal distributions with the probability of being pi and 1 minus pi. And Bayes R and S Bayes R will extend these two normal mixtures to mixture of point mass and three normal distributions. And more recently, PRSCS really becomes very popular by putting a continuous shrinkage prior on beta. So you can see there are really different choices of prior, but we often want to ask which one should we work on. And some of these prior assumptions also need to involve a large number of parameters to be estimated. And typically, one needs to use a validation dataset to perform the parameter tuning. And if we look at the real genetic architecture of the complex traits, you can see for traits like neuroticisms, the distributions is sharply peaked around zero, which means there are many SNPs with relatively small effects. But for other traits, such as the college completion, the distribution is really spreading out, which means there are lots of genetic variants with relatively large effects in the tail regions. And this means we cannot simply use a single normal distribution to describe this kind of distribution. And we often prefer to use a more complicated mixture of normal distributions to describe the genetic architecture of complex traits. And one thing I'm going to talk about first is the Bayesian process prior. So compare with these kind of the three mixtures or two mixtures models, it extends the number of the mixture components to a much large number. So in theory, this number should approach infinity, but in order to fit the model in practice, we often need to apply some truncation to the max number of components. And personally, I often found setting a large number like 1,000 will be sufficient to fit this kind of model. And this is just a Bayesian hierarchical model of the Bayesian process prior. And if we simulate our data to follow either a single normal distribution or a mixture of three normal distribution, and then we fit the Bayesian process model, we can see that the fitted density actually match the theoretical density pretty well. So this means the Bayesian process prior is really adaptive to diverse genetic architecture. The application of Bayesian's process random effects model can be first dated to 2010 by a paper published by Cassiller. And in 2017, Xiang's group published the papers extending this kind of process model to the genetic predictions. And in 2001, my first graduate project works by extending this genetic risk prediction model using the Bayesian prior to use of the summary statistics, which I'm going to talk here. So one question you may be asking is, these are all kind of prediction problems, and we all know deep learning is very successful in solving the prediction problems. So why does this field not using the deep learning for the genetic risk prediction? I think there are three main answers. The first is the scalability. Because the training sample size is typically 10 to the 5, and the number of SNPs is typically at the millions level. So this kind of a large data set really makes computation very hard for a very deep neural network. And I believe that the modifications of the structure of the deep learning needs to be applied in order to train a feasible model for the prediction. Second is about the dataset availability. So Y is a phenotype and X is a genotype. So these are all very sensitive personal informations which typically are not available even if the study is published. Instead, often a study will publish the summary statistics, that is the results from a GWAS study. So this means we need to transform a linear model to the summary statistic-based model. The easiest way to think about it is to multiply X transpose divided by N at both sides of the equations. So this means we are performing a linear regression for each of the SNPs separately. And if we assume the, the genotype matrix has been standardized, then the denominator is simply N. And if we have the informations of the estimated effects in the summary statistics, and we can use this kind of information to construct a new likelihood assumptions. Here, beta is the true effect we want to estimate, and beta hat is the effects in the summary statistics. Here, R is X transpose X divided by N, which is just a correlation matrix for the SNPs. And in a homogeneous populations, the correlations among SNPs, they are fairly stable. So we don't need the X from the original study to obtain this estimate. Instead, we can use the public reference panels such as 1,000 genomes to obtain an estimator of the R. So this is an issue of the privacy and the summary statistics. And the third point is, even though the biological process is fairly complicated and there's lots of non-linearity, currently the human genetic studies finds there is a limited evidence to support there is a non-linearity in the in the GWAS study. And because of there's not so much linearity, the linear model is still is very successful in estimating, for example, concepts like variability, and genetic correlations. And one thing about the deep learning is, in recent years we see there's a huge success of the transfer learning. So transfer learning means if we have two data sets and we fit model on the data set one, and we can use the information learned from the data set one and to construct another model in the data set two and to improve the prediction in the data set two. This concept has been very successful, for example, in the natural language processing and in the computer visions. And let's think about the transfer learning in the genetic risk predictions. I personally, I feel there's also a similarity of the applications of the transfer learning in the genetic risk prediction. For example, we can transfer the prediction model in one population to multi-populations. One example would be if we have a data set from the European ancestry with a very large sample size. But for the East Asians and African populations, we don't have a large effects training data set available. Then we can use the transfer learning to transfer the prediction model built in the European to the non-European populations. And the second example is the admixed populations. So admixed means part of the genomes, they are from, for example, for the African Americans, part of the genomes, they are originated from the European ancestry, and the other part of the genome, they are originated from the African ancestry. So the kind of hybrid scenarios means we can construct the transfer learning model for the different segments of the genomes based on their ancestry background. And lastly, suppose we have two traits that are correlated and have shared genetic information, we can use transfer learning to build a joint prediction model so that the predictive power will be higher compared with building a prediction model for a single trait. And this is our demonstration of the problems of the translating of the genetic risk predictions from the European to the non-European. First, most of the GWAS study or the training data currently, they are coming from the European populations. And you can see the red one is a European, and this is a sample size of the European GWAS compared with other colors, those GWAS being down in the non-European populations. And if we take the prediction accuracy of the European populations at a reference level, we can see that the prediction accuracy of a predictive model in non-European populations actually decays as compared with the European populations. This is largely due to the lack of the training data to construct such a prediction model in the non-European populations. And we can think about a way to transfer the prediction models from European to non-European populations. And the central idea for my research is to build such kind of the two-dimensional digital process random effects model. This model is a more general form. It assumes that we have two data sets available, and we assume that there's kind of four components for the prior assumptions of one SNPs in two data sets. So the first component with probability being P0 means the data, the effects, they are zero in both data sets. And the second component means the effects is non-zero in data 1, but zero in data 2. And we further use the Dirichlet's process to specify the mixture of the distribution for the dataset 1 effects. And for the third component with probability being P2, we assume that the effects, they are zero in dataset 1, but non-zero in dataset 2. And the last component means the effects, they are correlated and non-zero in both datasets. So suppose we don't have the last part, then it means there's no shared information being captured between two datasets. And it is equivalent to fit a single dataset model separately in the two datasets. But with the last component and with this correlation parameter rho in the model, then we can really capture and borrow the shared information across two datasets to help improve the prediction. And this is a general summary of this kind of model. So we explicitly model both the dataset specific and dataset shared effects. And importantly, we really borrow the shared information across two datasets. So if the dataset 1 have a much larger sample size compared with the dataset 2, and we can capture this correlation, then we are effectively translating such information from dataset 1 to dataset 2. And this is kind of non-parametric models since we are using Dirichlet process, so there's no need for parameter tuning. And this kind of model is typically fit by using Gibbs sampler and using Markov chain Monte Carlo. And more specifically for in this cross-population prediction settings, we are able to combine this kind of the prior with the data set from the two populations. That is the GWAR summary statistics from the one population of the European and another populations of the non-European. And we often we also take the reference LD matrix from the two populations. After we fit the model, we are able to output the effect size, estimated effect size for each SNP so that we can derive a prediction model based on this. And in this project, we compare our newly developed dishes process model with some of other methods. For the first three methods, they are all based on single populations, which means they only use the data from the non-European populations to fit the model. And for the last two, they are multi-population methods, but their assumption they made is a little bit different from ours. So XPath assumes the snap effects follows by various normal distributions into populations, but it does not assume that there is a population-specific terms or the zero terms. And the PRSEX assumes there's is more extensions of PRSEX to multi-populations. And the difference between PRSEX and with our model is we explicitly will use the will set up a parameter row to capture the genetic correlations. And this is the data we use to to train the different models. We select a bunch of the traits, and also we take the public GWAS summary statistics from the European, East Asian, and African populations. So you can see here I list the sample size of the GWAS studies, and the sample size is often much larger than the European compared with the East Asians and African populations. And in order to evaluate the effectiveness of the prediction models, we typically test the predictions in a large biobank. And in this project, we mainly focused on the UK biobank. So UK biobank is a population-based cohort consists of about 0.5 million individuals with their genotype data available, and also the electronic health record and lab measurement and some basic basic measurement like height and BMI. So what we do is we use the public data to train the prediction model, and then we apply the prediction model to predict the value, for example, for height in the UK biobank based on the genetic informations. And then we correlate the predicted value of the height with the real observed value of the height to measure the prediction accuracy and to compare the performance of different methods. And we first look at the East Asians, and we show the prediction accuracy in terms of the square of the Pearson correlations for these six representative traits: height, BMI, HDL, LDL, total cholesterol, and the triglycerides. So the red methods is our methods, and other methods they are in other colors. And we can see, generally speaking, we have about 20% improvement over other methods by applying this kind of model. And similarly, for the African populations, we are able to also see there's improved genetic prediction performance. And still, we focus on six continuous traits plus one binary traits, the type 2 diabetes, and we measure the predictive accuracies of these traits for these methods. And then we are going to talk about the admixed populations. So here I'm going to give a brief introduction of the admixture process. So suppose one parent is from the African and the other is from the European. The first generation will inherit one chromosome from each of the parents. So one chromosome will have the African background and the other from the European background. If after many generations, because of the meiosis, the recombination during the meiosis, and part of the genome will have segments originated from the European ancestry, and part of the genome will have the segments originated from the African ancestry. And this kind of the local structure of whether a segment on the genomes from the African or European background is often referred as a local ancestry. And we are able to reliably infer the status of local ancestry of any individuals based on our reference data. And a unique challenge of dealing with admixture is compared with the populations such as European and East Asians. If we look at the PCA plot of the genotype, you can see there's a continuous variations along the PCs. So this typically means the local ancestry will varies hugely across different individuals. For example, some people may be 80% of African and 20% of European, and others may have 20% of European, African and 80% of European. In order to handle these kind of the local ancestry informations, we can think about how do we model the effect size in these scenarios. So one way is to assume that the effects they really differs across different ancestry backgrounds. So here is a linear model links the ancestry specific genotypes to the phenotype. And we are interested to estimate for example, the African specific effects and European specific effects. And on the contrary, the traditional GWAS model assumes that the effects they are same across two ancestry. So beta J1 equals beta J2, so that they do not use information from the local ancestry. And we combine the local ancestry informations with our prior of the four components to formulate a model called SDPR ADMIX to construct a prediction model for the ADMIX populations. And we compare our methods with four other methods. First, the BSLM and IPGS, they do not use the information from the local ancestry. And BSLM is a sparse mixture of two normal, like a spike and slab model. And IPGS is basically elastinin-based model. And for our two other methods recently developed, Gaudi and partial PRS, they use the information from the local ancestry, although does not use a type of prior like we do. And one thing about Gaudi and partial PRS is they only use the part of the SNPs, not the genome-wide SNPs, because of some computational issue with those methods. And we train our prediction model based on the All of Us datasets. So All of Us is an initiative announced during 2015 State of the Union address and is run by NIH. And it has currently recruited about 0.6 million people with their genotype data and electronic health record data available across the United States. And the one features of the All of Us dataset is it has a large number of the endemic populations, which is traditionally not studied in a GWAS study. So we train the prediction models on the All of Us, and then we test the prediction accuracy in the Yuko Sorry. And we compare our methods, SDPR ADMIX, mainly with two other methods, IPGS and PRS. We are not able to compare with Gaudi on this dataset due to some computational issues. And we are able to see that for the four traits, height, BMI, platelet counts, and white blood cell counts, our model is able to achieve a 10 to 400% of the improvements of the prediction accuracy. And we further look at how our prediction model looks. We plot the estimated genetic effects of the African ancestry on the x-axis and the European ancestry on the y-axis. And you can see for different traits, the genetic architecture is really different. So for height, BMI or platelets, you can see there's lots of shared genetic effects between the genetic effects. They are correlated, most of them are shared between the European and African ancestry. But for the white blood cells, there are many strong ancestral specific effects. And these two SNPs, they are located on the chromosome 1, and they are in high LD with Duffy SNPs, which is known to have a significant impact on the white blood cell counts, the neutrophil counts in the West African populations, because it has some positive selective effect on the resistance of the malaria infections. So this variant have much higher allele frequency in the African populations, and this variant codes a chemokine receptor. And because this is a nonsense, it's a nonsense variant, so the deletions of this receptor will dramatically reduce the counts of the white blood cell. And we also want to look at how we can construct a prediction models in the ethnic populations of the Hispanic and Latino populations. So compare with African Americans, Hispanic Latinos, they are more complicated because they typically involve the admixture of three ancestry, the European red one, the African green one, and the Native American the blue one. And if we look at Hispanic or Latinos, you can see that generally two of the three ancestry that are often predominant within certain ethnic groups. For example, in Mexicans, the European and Native American are often predominant. And in the Caribbean countries, such as Puerto Rico and Dominico, the African and European, they're often predominant. So this means we can typically use the two out of the three ancestry as representations and just apply our previous two-way models to build a prediction for the Hispanic Latinos. And this is the results. Again, we test on the full traits and we compare with two other methods. And we can see that the prediction accuracy is about 22% over improvement over IPGS and about 100% improvement over partial PRS. And last, I'm going to talk about how to apply this transfer learning into cross-trait genetic predictions. So two traits such as schizophrenia and bipolar, they have genetic effects that are shared or have high correlations, as often estimated by the so-called LD score regression or RIMO approach. And we can just similarly apply this kind of model by taking the data sets from the two traits to correlate the traits and one LD matrix into the combination with our two-dimensional delicious process prior assumptions and to fit such kind of model to output the effects. And we are able to show that the newly proposed methods called Pleo SDPR is able to improve over other predictive models, include our previous developed SDPRX and PRCSX. Yeah, so this is mostly my today's talk, and I would like to thank my PhD and postdoc advisor Hong Yu for their supervisions. So this is most of my work is from my PhD and postdoc, and also thank my co-authors, and also thanks their data resources, which provides the data, training data, and testing data for this project. And thank you, and welcome for if you have any if you have any questions.
Yeah, thank you so much for the really great talk. Yeah, it's really inspiring. And I will maybe ask the audience to ask question first, and then our host can ask a question later on.
Okay, I think I can probably start. Thank you, Geyu. It was great. That you started from very beginning of the methods. It's a great opportunity for me to learn the field. Mike, I have a very naive question here. Now you have the models from a European population and then also the model from African population or other models. And can you by comparing the like parameters you have from pure European population and pure African population, you can you find what the difference, individual difference, like a SNPs level difference between African population and European population, for example, and then somehow going back to the like a molecular level of the difference, understanding of the difference, to kind of try to connect statistical analysis to more close to my area of like a protein or gene level or like a pathway level understanding of the biological difference? Is that possible?
I think that's a very good point. So first, at a genotype level, we can imagine the genotype or the genetic variation first is very different, and also the allele frequency are very different. So if we are talking about how the genetic effects are different, for this project, I didn't do this, but for the ADMIX project, I did. I actually look at how those estimated genetic effects differs across the two ancestry between the European and African. I would say most of SNPs most of the genetic effects showing difference, they are because the allele frequency they are very different. For example, if a SNP they have allele frequency around 0 in the European, but probably higher in the African, then we are just not able to estimate those genetic effects for the European. So that's why we'll see the model will assign it as the highly ancestry specific effects. But for common SNPs, I think also from the biological point of view, people are more inclined to believe that the genetic effects they are same across populations because the biology should be same across populations. So that's why for most of the common SNPs, and we are able to see their genetic effects, they are highly correlated or shared across populations. One example would be the white blood cells because this is a trait with immune-related function. And white blood cell, the Duffy null SNP is the most well-studied SNPs that has strong population-specific effects because of their resistance to the malaria reinfections in the West African, so that it has a positive selection in the populations.
Okay, yeah, thank you.
Thank you.
I want to quickly follow up with Dasuki's question. That's very interesting question to think about, like a biological validation of that. I feel like for the GWAS type of analysis, as Gogi mentioned, mostly they will see a allelic difference across ancestry, which we do not have enough power to detect those signals in the other ancestry. But on the other side, for example, for the gene expression, we see, for example, in the immune response, we see the different immune response of the COVID across ancestries, right? For those type of, we have single cell data available, and we are able to detect those EQTL, different EQTLs across ancestry. That's feasible. But my question for you is like, how to validate? I don't know.
I think I think even for the single cell, like EQTL, there's paper comparing the EQTL estimated for in the European and African, and their conclusion is actually the effects they are same. And to connect with the overall immune response, I feel it's more complicated. For example, we know that the immune, for example, like if you are linked with a blood cell count, then there are some strong ancestral specific SNPs or the or the variants that undergo selections in African populations. So that may explain a little bit difference. But more generally, you may imagine the genetic variants of the European and Africans, they are totally confounded with the environment, right? So this means you need to collect more data in a balanced study design in order to really separate out or control for the environmental effects to make such conclusion.
Oh, okay, good. Thank you. Yeah.
Hey, may I ask a kind of more maybe technical question? This was a great talk. So, um, how first, how many of these like SNPs are generally utilized to predict a complex trait, let's say like BMI or height?
So we typically use so-called head map 3 SNPs, about a million SNPs, and I think not most of them they are in the non-coding regions, and that's typically for the GWAS where people find the associations mostly coming from the non-coding region, not from the coding region.
And then that's fair. And then how close, if I may ask, the prediction gets? Let's say like the height that people, is it within a few inch of a height difference? Is it like a feet of difference? I mean, like how close we are thinking in terms of like real tangible outcome of these models.
Yeah, so there's a recent paper in 2022 which builds a prediction model based on 5.6 million people, and their prediction accuracy is about 40%. So this is, I would say, close to use the average of their father and mother's height as a predictions. And if we talk about the upper bound, then it should be equal to the heritability, which is 80%. But currently, we are still not able to achieve that. And one thing about how many inches are there, I think we often scale the prediction model to predict the predicted value to on the scale of 0 to 1. So we need to scale it back to in order to get a really sense of how accurate to convert it back to the original scale of inches. And personally, I haven't really looked into this issue myself, but I guess maybe at a range of 10 to 20 or something like that, because currently you can think about if the accuracy is at about the 30 to 40% at most.
Yeah, to me, always like these predictions, like I'm having very hard time to connect back the accuracy, even at 90% accuracy, to like what is the range or what is the real kind of tangible numbers that my grandma would understand as well. So that's where I'm sometimes having difficulty of like when we talk about BMI, let's say, what is the range differences that these predictions are completely missing, and then how much room for improvement there are?
We can definitely convert those prediction model, the predicted value into the original scale, and it doesn't affect the correlations or the accuracy.
Fair. And then do you think that additional modality besides variations are also having effect on these predictions?
Yeah, certainly the environment will have lots of impact. For example, the impact of the diet on BMI, and also epigenetic factors will also play an important role. And I would say one interesting research direction is to integrate all those information together to improve the prediction.
Yeah.